Bismillahirrahmanirrahim, and welcome back to this next video. And this is another video in the series of videos on the bioinformatics. In this particular video, I want to focus on the structures of the RNAs with their sticky strands. When you talk about the RNA molecules, we usually say that these RNA molecules, they consist of single strand of nucleotides, but they have got a natural urge for pairing with their complementary sequences, just like the DNA molecules. Now these single-stranded RNA molecules, they pair different regions of their sequences to form the stable double helical structures, much like the double helical structures of the DNA. Now why these stable double helical structures are required for these single-stranded RNA molecules, that will be clear in a while. So once these RNAs, these single-stranded RNAs, they are synthesized, each RNA molecule that quickly adopts a compact fold, trying to pair as many nucleotides as possible to get a stable double helical structure. Now these hairpin shapes, they are one of the basic elements of the RNA secondary structures, and these hairpin shapes, they usually consist of two parts. One, they are known as the loops, which are actually the unpaired regions of the RNA, and the stem, which are actually the paired regions of these RNAs. For example, this one is the structure of a transfer RNA, and I hope you know the functions of the transfer RNA, that they are one of the important component in the process of the translation, thereby helping in the synthesis of the proteins in the cells. So if you look at the structure of the transfer RNA, as you can see over here, this is one loop. As you can see, these nucleotides, they are not pairing with each other. This is another loop. Again, they are the unpaired nucleotide sequences, and you can see over here, this is another loop. So you have got these unpaired regions in the RNA molecules, and these unpaired region, they are known as the loops. The stem region, as you can see over here, these complementary sequences, they are interacting with each other, thereby making a double-stranded helical structure in the RNA molecules. And the combined effect of these loops and the stems, they actually help the RNA to perform their functions in the cells. So these RNA molecules, you can expect these loops, the unpaired regions, and you can expect these stems, the paired region in the RNA molecules. Now the RNA chain, they folds in a specific 3D structure which is dictated by the sequences. As I've told you in my previous video, that the structure of a protein is dependent on the sequence of the amino acids, and the sequence of the amino acids is actually dictating the 3D structure of a protein and hence its function. So the same is true for the RNA sequences as well, that these RNA sequences, they fold in the specific 3D structures, and these 3D structures, they are dictated by the sequence of the RNA. And these specific 3D structures, they are actually responsible for the biological functions of the RNA molecules. So this 3D structure, as I've told you, in the form of the transfer RNA, which is an important component of the translation. So if the transfer RNA assumes a specific 3D structure, it can help in the process of the translation. Otherwise, it cannot help in the function in the process of the translation. So this biological function of the RNA molecule, that will be derived from the 3D shape or from their sequence complementarity with the specific genes. One of the important thing in this current era is that recently it has been discovered that the small RNA molecules can switch off the activity of a number of genes, that has triggered a renewed interest in these sticky RNA sequences and the 3D structures of these RNA molecules. We will continue the discussion in the next video. So if you like the video, please subscribe to my channel, hit the like button and share it with your friends.